Hello all, this is Dr. B. This is a intro tutorial for NCBI, which is from National Center for Biotechnology Information. It is a database of many things, most of which are public DNA, RNA, and protein sequences, annotations, etc. And so I'm going to go over a generic kind of overview of this, and then I'm going to talk about a couple aspects of it. And then later in the course, I may talk about some other pieces. So generally, you get here to this main page. You can see on the left, there's all kinds of resources where you can be more specific and look at any of these. We'll look at some of them later. Some of the ones you may find interesting would be the training and tutorials, which is here, and also here. So if there's anything that you want to try, or it might be a little different for your assignment than this particular video, you can go there and find them. But generally speaking, I just like to start off with a general example to kind of show you some of the fundamental things that it can do. So if I would look up something like, let's see, I know that in one of chapter 1 there was mention of Huntington protein or Huntington chorea. Let's go ahead and Huntington, which sometimes is also written as this. So this is a particular protein that has to do with Huntington disease. Let's just see what happens when I click on that. And when I click on that, it basically shows me everything in relation to some of the things on the left, and in a really nice ordered way. And so what are some of the literature? Now, PubMed, by the way, let's start here. PubMed is one sub-database of NCBI, and basically it is a collection of all kinds of peer-reviewed articles in relation to the topic at hand, and you can do all kinds of more specific things about it. It's really neat. So it's a great way for you to kind of find some good peer-reviewed stuff. This is what makes this site way better than like a Google Scholar or just Google in general, as you can, in most cases, be trusted that it's been peer-reviewed as composed of just somebody writing a web, article and telling you what this is. So that's what that part of it is. And a couple other things of interest. So in terms of individual genes, you're sitting there wondering, well, how can there be 734 genes for Huntington? And by the way, either one of these is appropriate. Huntington is the protein and Huntington is the disease. So that's a whole other thing. But let's just click on this and see what happens when we go here. And you're like, okay, wow, what's all this? And the reason that there's so many of those genes, you can see in the first example here, is that this one happens to be in humans. I don't know if you knew what mus musculus is, but that's a mouse. Actually, it says there, house mouse. So there's other organisms that also have the Huntington protein, and there's also versions of them called isoforms, and we'll talk about those later. So generally speaking, this would be some of the different ways you could find the genes. If you're looking for the human one, you can just literally click right here. And then this will give you some background on the actual gene itself. And so there's a couple things that kind of let you know. First is the lineage of how this taxonomically is ordered, a summary of kind of what it does. And you can see here it has the numbers of exons and kilobases of the gene and what parts of it are transcribed, all kinds of neat stuff there. And then there's some information down here. And one of the things this kind of does, you can see here location. So this is found on the human chromosome 4. That's what the 4 is. It's found on the P arm of the chromosome. Chromosomes have two arms, P and Q. And area is 16.3, and it has 67 exons, which basically means that there's 67 segments of gene coding DNA that are separated by introns in this particular gene. And then we move on to a couple other things, which is kind of relatively speaking where it's located. I'm not going to spend a lot of time on this part right now, but I do want to just talk a little bit about these three things here. And you're like, what is this weird looking line? And what are these little teeny things down here? And what's all this stuff? So let's just explore. Let me just hit the word graphics and see what we get. So if I click on graphics, then I can see again the same kind of deal, but it's a little bit more hands-on-y, a little more annotated, where I can move things around. I can look at different sizes of this. I can scroll in, scroll out, look at single nucleotide polymorphisms, which is this part here. This is where there might be a one-letter difference in some of these. And again, we'll get into all this later. This is just an overview video. This kind of gives you a overarching kind of view of what's in the gene and what has been annotated, which means identified as serving a purpose. So that's the graphics one. If I just go back and I hit FASTA, which is this, or sometimes called FASTA, this is the actual DNA of the gene. And this code right here is the reference sequence, and some people use it interchangeably with the accession number. It's basically the way that you would identify the gene through a number, and it's the only gene in the world that has that code. And so the first line is always basically that information, and you can see here that this is the actual gene itself, the whole thing. And if you look, I'm not going to go, I'm going to scroll down to the way right. I mean, it's a big boy. And that is all the DNA that codes for that gene. And so that's what FASTA is, or FASTA. And if I go back again here, and then I look at GenBank, that's the third one from this particular thing here. If we see GenBank, this is a little bit more information about this particular sequence. So the question is, how did that super long sequence get into this database that I showed you about 20 seconds ago? The answer is somebody submitted it to GenBank, and GenBank is the public record of all of the DNA sequences. So when we saw that there were 700 some odd Huntington protein or Huntington gene submissions, that means that they were all submitted by people that did DNA sequencing and submitted it. So a couple things you can see here is some really good information. One is this thing clearly is 169,280 base pairs of a linear chromosome. It comes from a human, chromosome 4. Here's the accession number, which is the ID number. There's a couple other things that link to where this is found. I'll talk about RefSeq later. It's found in Homo sapiens, and this happens to be the paper that the people that were involved in the paper that related to this sequence, and they are the ones that submitted this paper to GenBank. I'm sorry, not paper, sequence to GenBank. And this is the name of the paper that they wrote, and this is the journal it was in, and there's a link to it. You can see all the way down here, and this is obviously a lot of people. And as you get down to here, you start getting some other cool stuff. And this happens to be some of the features of this particular gene. And this is kind of like some parts or domains of what, in essence, what parts some of these would be. We also see here that here's the gene itself. And then we have a connection to the actual mRNA. So if I click on the mRNA, you can see here it kind of shows you this particular segment of mRNA that was identified and at least coded for from the DNA. So let me now go back, and you can see them all here. That's the coding part. And let me now go back real quick. So I had to do that because it was too big. So let me now go back here. That was a really long piece. So then we have what's called the CDS, which is that's kind of related to the mRNA. That's the coding aspect of the DNA. That's the part that relates to the introns. And if we keep going down, you can see here that this is the translation. If you recall from probably the Edpuzzle you did probably a couple of days ago or a few minutes ago, you know, translation is taking the messenger RNA and translating it into the amino acid sequence. And you can see here that here's the very beginning, and that makes sense because the first letter is methionine, and methionine is the first amino acid of most proteins by far. And so this is a lot shorter than that gigantic list of DNA that you showed, because you know, there are three DNA nucleotides for every one amino acid, and also this does not include any information from the introns. So this is just a generic kind of, these are just miscellaneous features that people have identified from this particular protein itself and some areas of interest of it. There's a lot of them. This is like about 1/20th the length. If I just scroll real fast, you can see there's a lot. And of course down here is finally the actual FASTA, or the DNA for it. So that is one little aspect of how I could have done that. And you'll see here, by the way, that this now says nucleotide. So there's a lot of different ways to get to the same thing with NCBI. And so that in itself, let's just kind of go back to, let's just do the very beginning and do all databases. And go here. I could have also just wanted to be interested in the nucleotide. And you're like, nucleotide? Why is there 43,157 of those when there only appear to be 7,000 genes for it? Well, if we click on it, nucleotide, this is a combination of every submission, some of which are just parts of it, some are which of the whole gene, some are mRNA. So you can see here that this is a clone, so that's probably in a plasmid. So these are every single one of the pieces up there. And you're like, wait a minute, I can't look through 43,000 of these. I need a more specific. So what you do is you would click on Advanced. Let's just do a little, like a different example to kind of show you. Let me just do P53. It's just a protein. And let me just hit that. And we can see there's 66,368 cases of P53. If I were to do this advanced, and then if I did, the gene name is P53, and if I do Homo sapien as the organism, let's see what happens to that number. I should add an S because it's a lot of us have it. So now that number goes down to 106. And you can see here that there's all kinds of different versions of what people have put in for different versions of this particular gene. And so if we just randomly click on, I don't know, this one, this looks good. This is just a piece of it. We can see here from before it has a name to it. We see what it is. It's a tumor suppressor protein. There's a variant to it. We can see all kinds of neat stuff here. And one thing I want to know is, well, what are the closest other sequences to this? If I scroll down here, there's my, all the annotations, there's the exon. There's that piece right there. Are there any other related organisms or sequences to that? I can just hit this thing called BLAST. This is new, and I'm going to do a short version of this now, then do a longer one later. But basically, it's going to compare that sequence to everything else in the database to see how similar or dissimilar things are. And this is BLAST N because I'm blasting a nucleotide sequence. This is for protein, so we'll go down the list of these later. So let me just hit BLAST. I'm not going to worry about any of the other things. I just want to see. And what's it doing? Right now it's comparing that sequence and finding similarity with any other sequence that's in the database. And so we would hope that there would be other human P53 sequences that would align with it. Sometimes this process takes anywhere from 5 seconds to sometimes 2 or 3 minutes. So I'm going to pause the video till it's finished. All right, there was really no need for me to pause. It only took another 5 or 6 seconds. So basically, this is another aspect of NCBI, and this is called BLAST. And basically what it did is it took the sequence and it compares it to other sequences in the database to see how similar they are in nucleotide alignment and basic length. And it typically gives me 100 at a time. And you can see here that the most, that right here, Homo sapien, looks to be a lot of the top ones. Gorilla makes sense because it's, very close related to us. But there's only one of them that's 100% the same and 100% the length, and it's this. So I'm going to turn this off right here, and you're probably like, what are you doing? And all I'm going to do right now is click on this, and I'm going to click on something called alignments. And basically what it's doing, which is really neat, is it's comparing basically these two here. So you see here that there's something called query and subject. And so the one that I submitted, the BLAST one, that's the query. And then the subject, this is what came back from the database that it said had the best match with. So, and you can see that they're pretty much all aligned because it's 100%. I'll talk about identities and gaps later. So let me do one that's not 100%. Let me just now click on this and do aligned. And we'll see here that it's saying that there's 2,596 out of 2,614 exactly the same, and there's also some gaps in that. So we can see, oh, there's one. You can see how there's no line between it. That's how we know that there's a different letter right there. And so if we go along this whole thing, we're going to find more of those. And this is one of the ways that we can see the relationship between how both of these relate to each other. The query, which is the thing that we're using to submit, and then the spit out, I like to call it, from the database, that kind of tells us which one it aligns with. And so that is one of the things I can do. I can also do a graphical summary, which is not a very interesting one here. I'll get to that later. And because I only chose one, we have basically a taxonomic identification. So if I go back to descriptions and I pull a couple other different organisms here, like that, and I hit taxonomy, it'll tell me the relationship between all of these. So this is just a short introduction. It's enough to be able to do your first assignment, as well as the web query that I created for you to do with NCBI. So if you have any questions on it, please let me know.